Scientists believe that gene editing is the future of medicine. It works by changing how an organism's DNA works by removing, replicating, or adding new genetic material to cure diseases that mankind thought were incurable for a long time. As this new technology emerges, researchers at UCL have found a way for it to treat resistant leukemia when they have exhausted all other options. But how does it all work? Stay tuned to find out. First up, revolutionary new technology might be the answer to resistant leukemia. Leukemia is a cancer of the white blood cells, or your bone marrow. Usually, the disease is treated through chemotherapy, like most other forms of cancer, but sometimes a major issue in the treatment of acute leukemia is the fact that the patients develop a resistance to chemotherapeutic drugs, leaving them with essentially no option to get better. But scientists at the UCL and Great Ormond Street Hospital for Children have come up with a potential cure, using engineered donor T cells to treat children who have developed the severe condition, published in Science Translational Medicine. This research is the first time gene editing has been used on such a large scale. For now though, the trial is only focusing on children to curb the disease in its very early stages. Up next, how does it all work? Phase 1 of the research and trial is in full swing. The process makes use of what the researchers call CRISPR-edited cells. For those of you who don't know, CRISPR is a form of gene editing technology that helps scientists alter DNA sequences and modify gene functions. Usually, CRISPR procedures have been used to research mental illnesses and cancer. But in this case, the CRISPR technique is used to make cuts in the patient's DNA to insert a brand new genetic code. Now, this new genetic code targets the patient's T cells. Your T cells are an important part of your immune system. Essentially, what they do is that they directly kill infected host cells and activate other immune cells to regulate your body's response to diseases. In resistant leukemia, your T cells begin to lose functionality, and that's exactly the point where the researchers from UCL want to target the disease. So, the genetic code helps your T cells express a reception known as the chimeric antigen reception, CAR. This essentially means that your T cells can then identify certain markers on the surface of cancerous cells and destroy them before they invade your body. What's really great is that these modified T cells have been gene edited even further. So, they are standardized and don't need any kind of donor matching for the patients, making the therapy even easier and more accessible. Followed by, is this the answer to resistant leukemia? All things considered, this sounds like a pretty great way to treat resistant leukemia, but the only roadblock is that the therapy requires doctors to collect and engineer a patient's own cells for now, and this process is pretty expensive and, in a lot of cases, not all that easy. In the words of Dr. Qasim himself, whilst there are challenges to overcome, this study is a promising demonstration of how engineering genome editing technologies can be used to tackle unmet health needs in some of the sickest children we see. So, to battle this struggle, the research team is looking into the idea of donated T cells to be pre-engineered and then used for multiple patients to cut costs. For now, the researchers at GOSH have manufactured their sample donor T cells with a single disabled virus to enable the CAR and CRISPR technology. All the donors were healthy individuals who volunteered to be part of the process. Moving on to the foundation of the research. While the research is definitely new and revolutionary, it actually builds on Professor Qasim's previous work. The only change is that the researchers now have access to much more modern technology. It all started in 2015 with a 1-year-old infant who went through a similar CAR T-cell therapy to help her body fight leukemia. The doctors used white blood cells collected from a healthy donor. They used a molecular scissor technology called TALENs, a predecessor to CRISPR, to transform the cells and give them the ability to destroy leukemia cells in the body. While the experiment was a huge risk back then, it was a success. And that's what led to the trial we're talking about today. At the same time, Professor Qasim said that his therapy is not a permanent solution for all kinds of leukemia patients. According to him, the procedure works as a bridge to keep leukemia patients alive until their matched T-cell donor can be found. Why is that? Well, because in this kind of therapy, the patient is destroying his or her own immune cells, which are then replaced by the modified donor cells. But at the end of the day, the best case scenario is for the patient to start producing their own T-cells, which is where stem cell therapy and transplant come in. Let's talk about the trial. The participants of the trial included 6 children aged 14 months to 11 years, all of whom had been treated for leukemia, but the procedure simply hadn't worked. These children had been through all standard treatments for the disease, but had unfortunately relapsed multiple times. As part of the trial, the edited cells were transplanted within the children's bodies, and then the researchers gave the entire experiment 4 weeks to switch into action. What they hoped was that in 4 weeks, cancer would be drastically reduced, and if successful, the patients would then be put through a bone marrow transplant to boost their immune systems while also managing any side effects in the process. What's really great is that 4 of the 6 participants entered the stage of remission within 28 days, where their cancer was drastically reduced. This helped the doctors kickstart their stem cell transplant, and while 2 of these 4 children sadly relapsed after the transplant, 2 of them are now in remission for 9 and 18 months respectively, which gives the doctors a lot of hope. According to Professor Vijay Arora, a leukemia specialist at GOSH, the children treated in this study were facing the worst possible prognosis with their disease. It is only possible for us to save more young lives through clinical trials, and we are forever grateful to all the families who have been involved in this study that will help more children in the future. Coming up, why is this research so important? The reason why this research has proven to be so revolutionary is the fact that the survival rate for leukemia is less than 10%, and the number goes even lower with more aggressive forms of the disease. In this particular study, the scientists focused on chronic myeloid leukemia, which essentially has no cure when it's in an acute or blast phase. According to Jivisha Bajaj, PhD, assistant professor at biomedical genetics at the University of Rochester Medical Center, and a researcher at the Wilmot Cancer Institute, this kind of work is particularly important for the discovery of new treatments. These genome treatments pave the way for doctors to understand how cancers grow on a deeper level. In the future, with the introduction of other technologies, scientists will be able to study cancer microenvironments and the area around the tumor to see exactly how the disease behaves and where it can be stopped most effectively. Lastly, growing concerns. According to Dr. Qasim, as effective as the treatment can be, it does come with its own risks, with the most concerning one being G mutations and cuts. In addition, according to the researchers, the DNA-delivering vector can actually remain in the body for years after the initial injection, and that could have some unexpected responses. For example, the body might have an immune reaction to the DNA-cutting carrier, which might interrupt the entire process. Another major concern is the fact that getting the vector to actually edit the target cells instead of working on others. According to Qasim, ensuring that enough target cells are edited and the vector delivers its payload to the right part of the body is one of the biggest challenges in the research. If this fails to happen, the patient's bodies can develop a number of disorders and abnormal immune responses that can actually worsen their case. All in all, as great as the treatment is, it might still take the research team years to figure out how to actually make it work without putting their patients at risk. And the only way to do that is through rigorous testing and trials. With the trial proving to be so successful in its initial stage, doctors believe that this study is only going to add to the currently available treatments for leukemia. For now, the next step is to include more and more participants in the research to make it more foolproof and effective in terms of treatment. Hope it works out. That's a wrap for today's video. Do you think gene editing really is the answer to cancer? And does this therapy actually sound promising? Let us know in the comments below. Make sure to give this video a thumbs up and subscribe to our channel for more videos like this. See you in the next one.